All right. I think we can go ahead and get started. Thanks everybody for joining us, and I'm sure there will be more attendees starting to trickle in, usually the case. This week, this is part of our Vincere's weekly journal club, which is part of our open access or our open science strategy. We know that science does not happen in a vacuum, and of course, having the experts talk about the beautiful work that they do is always nice. So thank you, Dr. Navikas, for being here. As you can see, we're joined by Dr. Shohreh Issazadeh-Navikas, who is professor at University of Copenhagen, interested in neuroinflammation and neurodegeneration, which obviously overlaps with our interest here at Vincere, and I'm sure many of you out there. Today, she's going to talk about her team's recent manuscript in Molecular Psychiatry, which is titled Mitochondrial DNA Damage Triggers Spread of Parkinson's Disease-like Pathology. And we'll keep it brief for the intro, so we have time for questions afterwards. And with that, I will hand it over to Dr. Navikas. Thank you again for joining us.
Thank you very much for the invitation, for the kind invitation and the chance to share our results with you. I, in advance, apologize for my voice. I have bad cold, so I hope it will bear with me until end of the discussion. Yes, so since I am not sure about the background of the attendees, but as I understood, you have quite much of overlap interest, but nevertheless, I will mention that one of the diseases that we are interested in our lab to understand more molecular mechanism of the disease is Parkinson's disease, which is the second most common neurodegenerative disease. Majority of the patients suffer from so-called sporadic, so there is no specific gene associated with it, it has been found, but majority are developing motor symptoms in particular, but again, over 80% by time they develop also cognitive impairment, which has received less actually attention in the field. And currently there is no cure. So the disease mainly, it is believed to impact substantia nigra and midbrain, but again, although the most affected neurons are dopaminergic neurons in substantia nigra, the disease is highly spreading and it is affecting basically most of the brain. It is associated with neuroinflammation, and the major hallmark of it is accumulation of aggregated phosphorylated alpha-synuclein in Lewy bodies, but also they contain phosphorylated tau, phosphorylated A-beta, ubiquitin, name it. And this is because of the blockage in lysosomal autophagy pathway that normally should take rid of the waste in the cells. The disease is well known to be associated with mitochondrial impairment and accumulation, while the reason behind it is less understood. So major hallmark of the disease are coming by aging. It is containing of neurodegeneration in broad sense, aggregated phosphoproteins because of lack of waste protein degradation, mitophagy causing mitochondrial dysfunction, and also neuroinflammation. So another background to the paper that I know you are interested in, the journal club of it, is that some years ago, we stepped into a finding, or a very interesting finding, that neuronal defects in or lacking a cytokine, antiviral cytokine, which is also neuroprotective, interferon beta, or its receptor, IFNAR1, result in actually a spontaneous Lewy body pathology and outcome very similar to Parkinson's disease dementia, and that is because also they lack or they are defective in autophagy. So this is also was followed by finding that indeed transcriptomic changes of the brain with PD patients, especially PD patients as they develop to dementia, it's highly associated with type 1 or interferon beta IFNAR signaling pathway disturbances. And this was causing also mitochondrial dysfunction in the patient as well as the models that we were showing. Furthermore, lack of neuronal IFNAR is causing mutation and alteration in mitochondrial DNA, as well as it is important for rescuing the neurodegeneration because it is important for a regulation of mitochondrial fission upstream of mitophagy. This is causing, indeed, so each color is one mitochondria in EM of neurons, and this is a corresponding one in if they lack interferon beta signaling. They become gigantic, and they are basically blocking especially axonal function. So here today, the journal club you are interested in, we were asking, as most of unknown pathway in neurodegenerative diseases, what is actually initiating and causing the spread of Parkinson? Does it have anything to do with mitochondrial dysfunction and damaged mitochondrial DNA that we have observed? So indeed, again, transcriptomic analysis of cohort of patients with Parkinson disease showed highly dysregulated oxidative stress pathway, DNA damage binding, cytosolic DNA sensing, and TLR pathways in patients. And in another cohort, we could see indeed that ND4 and ND5, which are part of the respiratory chain of complex 1, are significantly deleted in patients and also in mice model that we are describing here. I think I will close my window here to be able to see the slides. Okay, next we were looking at the neuronal cells by immunofluorescent staining of mitochondria and for oxidative stress, 8-OHDG, which showed indeed significantly increased level of oxidative mitochondrial DNA in these cells. And again, by sequencing, we found massive mutation, deletion, insertion in mitochondrial DNA, but mainly in a hotspot around ND4, ND5. Okay. Yes. So next, we asked the question, what is the fate of this damaged mitochondrial DNA? What happens to them indeed? If we look in cells again by immunofluorescent staining, staining for DNA, as well as a mitochondrial protein here, HSP60 or any other like TOM20. Interestingly, in normal wild type cells, here we are looking at MEFs because they are easier to image, but in the supplementary, you can find also in neurons. Normally, mitochondrial DNA, it is in mitochondria, as it should be, while if they lack interferon beta signaling, they extrude it. We could see quite often mitochondrial DNA outside the mitochondria, significantly higher in knockouts. And by electron microscopy of brain, we could see indeed quite often they are having vesicles extruding from mitochondria, again, in significant amount in neurons that lack interferon beta signaling. They also extrude this in the supernatant. So if we take supernatant of these neurons which lack interferon signaling, we can see a quite high number of or copy number of mitochondrial DNA in the supernatant, and they are oxidized similarly. And also, if we purified extravesicles from neurons, they also contain high level of mitochondrial DNA. This is also the case if we look at 6 weeks of age in the brain of mice lacking interferon beta. And this, just to highlight, indeed, it is much earlier than any other pathological hallmark of the disease show up or any clinical deficits has appeared. So next we asked, is damaged mitochondrial DNA that we see here pathogenic per se? So if we purify mitochondrial DNA from normal neurons or from those that they lack interferon signaling, so interferon beta knockout or receptor knockout, and inject them specifically in forebrain or substantia nigra and stratum, and start checking clinical outcome, behavioral outcome of these mice. 2 weeks later, they already start showing disturbance in motor coordination significantly. They do exhibit neuropsychiatrical behavior like anxiety by excessive climbing and more climbing events, as well as delay in finding the exit box in a cognitive test called barn maze. And this is associated with, indeed, dopaminergic neuronal death, both in stratum that I don't show here, but also substantia nigra. So this is normal control group that receive either nothing or wild-type mitochondrial DNA or mitochondrial DNA from interferon knockouts mice. And as you can see that they are significantly losing dopaminergic neurons, and they are having increased caspase-3 dependent pathway of cell death in the brain. And this is associated with accumulation of phosphorylated aggregated alpha-synuclein, another hallmark of the disease. And indeed, 3 months later, if we look by EM, both that they have received interferon beta knockout or damaged mitochondrial DNA from them or from receptor, they also develop Lewy bodies with as well as all sort of lipofuscin granules and inclusion bodies. Indeed, this is also associated with neuroinflammation in the form of astrogliosis, but not much of microglia activation. And furthermore, that is causing also oxidative damage assessed by oxidative DJ1. Sorry. So the next question, I'm sorry. The next question we were interested to address was, can this damaged mitochondrial DNA also spread and cause the progression of the disease? As I mentioned, these models are progressive, similar to patient. So we have injected the damaged mitochondrial DNA in cortex or in midbrain, while if we look at olfactory bulb, indeed we find, compared to controls, we find high levels of caspase-3 positive neurons in the brain, showing neuronal cell death, the Western blot of them showing same, also completely the caspase activation has taken place and oxidative stress is increasing. And this is interestingly associated with the dopaminergic cell death in olfactory bulb. What I do not show here is also cerebellum, which is quite far distance from the injector site, but the pathology has been seen also there. So, of course, the next question of interest was, what is the mechanism by which they can cause this damage? Mitochondrial DNA can cause disease. As mentioned earlier, one of the pathways that is significantly dysregulated in patients is TLR signaling. So, and among them are TLR9 and TLR4, which we checked in the cohort that we are having in collaboration. And we could see that indeed TLR9, it's downstream IRAK1, as well as TLR4 are significantly increased in PD patient, and it is the same if we look at brains of mice that have been injected with damaged mitochondrial DNA from IFNAR lacking mitochondria. And that is the same with neurons that receiving mitochondrial DNA. So the next question, of course, was what is the role of this TLR4 and 9? And indeed, what we could see was that if we block TLR9 specifically, the oxidative damages that has been significantly increased by injection of the damaged knockout mitochondrial DNA is being rescued. However, the cleave caspase 3 positive neurons, so neuronal cell death, is not rescued by blocking TLR9, and neither by blocking CGAS pathway, which has been shown as one of the major pathway associated with mitochondrial DNA damages. While if we block TLR4 pathway, that basically rescues the neuronal cell death completely and increases their viability. So more interestingly, we wanted to understand how this damaged mitochondrial DNA are being extruded and being recognized. So is there any molecular target that we can find that is responsible for a spread of the pathology? And interestingly enough, by purifying extra vesicles from damaged basically neurons that they receive knockout mitochondrial DNA, we could see that they can trigger, similar to pure damaged mitochondrial DNA, they can trigger neuronal cell death. And the proteomics of them compared to wild type showed heavy burden of mitochondrial DNA associated proteins or mitochondrial or mitophagy associated protein downregulated, while sensing of DNA damages have been increased. And interestingly enough, among the highest pathways associated with the disease were mitochondrial DNA maintenance, oxidative DNA binding proteins, and TLR ligands. Combining or overlapping these differences end up showing only one protein in common, and that was ribosomal protein S3, and that it is the only, very little is known about it, but at least it was known that it's binding to oxidized DNA. So next we wanted to understand if this is involved in the pathology. And indeed, if we purify brain extravesicles, they are enriched in RPS3 in these extra vesicles compared to normal wild type. And also, if we look at the cellular level and fraction neurons for mitochondrial contents and cytoplasmic, they are quite enriched in cytoplasmic in knockout neurons, but in wild type, they are basically similar. So that tells us that basically they are extruding it to the cytoplasmic area, which by doing immunofluorescent staining of MEF cells, we could see indeed that RPS3 and mitochondria are co-localized, and in knockout MEFs, basically they are extruded mainly. So although there is much heavier load of RPS3, they are much less in in the mitochondrial proportion, but they are mainly extruded extracellularly. So does this RPS3 bind directly to damaged mitochondrial DNA, and can it be involved in the pathology? Indeed, we could, by triggering using H2O2 to induce mitochondrial damage in N2A cells, see high level of mitochondrial DNA binding to RPS3 that we could co-IP and then do qPCR of them to show that hugely they are increased, as well as in neurons that they lack the signaling. RPS3 also, it is highly increased in neurons, normal neurons that are challenged with knockout mitochondrial DNA, that they are damaged. And if we co-IP, we could co-IP it with TLR4, it comes down, so RPS3 directly binds to TLR4, which has been known as an activator of TLR4. So next, we wanted to see if it's functional, and indeed, if we knock down the RPS3, the H2O2 induced extrusion of mitochondrial DNA can be completely rescued. So it requires RPS3 for damaged mitochondrial DNA to be expelled. And same if we knock down the neurons that they receive damaged mitochondrial DNA, they also rescue, not being able anymore to extrude mitochondrial DNA. And this is also rescuing cellular viability and blocks basically neuronal cell death. So to conclude from what we have shown in this paper, so in addition to what I mentioned about the role of type 1 interferon in patients, what we believe that we can conclude out of this study is that indeed neurons that they lack interferon beta or its receptor, so they lack the signaling, are incapable to regulate mitochondrial biogenesis. This is causing high oxidative stress to damage mitochondrial DNA, which is binding to RPS3 and being extruded most likely in EVs. But it can also, at least we had evidence that alone mitochondrial DNA, purified damaged mitochondrial DNA, can also cause pathology. Upon release of these extra vesicles with the damaged mitochondrial DNA and RPS3, they are most likely being uptaken by neighboring neurons or even neurons far distant from their origin. And upon release of the complex in endosome, they then being recognized either by TLR4 or by TLR9 through directly damaged mitochondrial DNA, or vice versa, RPS3 binding to damaged mitochondrial DNA. And in sort of activation, both TLR4 and TLR9 are causing the pathology we see, including TLR9 being responsible more for oxidative stress, and TLR4 pathway more preventing neuronal cell death, or its pathological activation causing neuronal death, and astrogliosis and neuroinflammation associated with disease and full-blown basically progression of the disease. So in take-home message, yes, damaged mitochondrial DNA can initiate indeed the PD-like pathology in the brain. And it's not only sufficient for initiation, it is also spreading an infectious-like pathology throughout the brain, and this might be behind the progression to actually dementia. Hereby I stop. Thank the group that are behind this work, and we'll be very happy to take any question you might have. I will stop sharing so I can see the audience if I can.
Wonderful. Thank you for the presentation. So nice to hear it from you directly and be able to ask questions. And there are already several questions in the chat that I'll ask. One question, and I'm kind of modifying the question from Andy in the chat, because we recently, actually last week, we had a talk by Laurie Sanders looking at mtDNA lesions in the blood of patients with Parkinson's disease, which I think matches what you're saying. It's all along the same theme.
I have seen the paper.
Yeah, I wonder if you can talk a little bit about copy number, mtDNA copy number versus mtDNA damage or lesions in what you're seeing.
Yeah, so actually that was one of the things in the press release we were hoping, and then I came across that someone else already happily did that. So the copy number, I think they also addressed it in the paper. There has been several previous studies showing damaged mitochondrial DNA associated with PD, but also quite conflicting. It can be both because of the technology, maybe not sensitive enough at the time, as well as the areas that they have been used, the stages of diseases, and so on. And we were hoping to see something like in the blood, because that might be a very excellent and a strong tool, actually, to see if it is associated with different stages of the disease. I am not able to address more the previous work because I haven't read it through yet, but it would be, of course, very valuable if that can be associated with developmental stages of the disease to dementia, to see if there are any specific mutation or oxidation that are associated more or less with the disease. I think that is another issue we have today. We can see a huge number of mutation, deletion, insertion, as well as oxidation, but it is with the current knowledge, we don't really know if there are some of them more important than others and how they are involved.
Makes sense. Makes sense. I'm going to squeeze in one of my own questions in between the other questions that are in. Did you look at other, I know you looked at MEFs and neurons here, but did you look at different neuronal types? We know that there's differential susceptibility within the neuronal types in the brain. Did you look at and see if there was degeneration in any of the other neuronal populations, both susceptible ones and maybe other resistant neuronal sets?
So yeah, it's an excellent question. So in vitro, we have looked into MEFs and 2As as well as cortical neurons. But in vivo or ex vivo, we have looked basically at different areas and regions of brains. And that is one of actually the major take-home message, is that although we inject damaged mitochondrial DNA in particular areas, this is high spread. So it does also cause neurodegeneration in cortex and in areas that they have not received damaged mitochondrial DNA, distance from them. So in olfactory bulb, in addition to dopaminergic neurons, in general, we could see high levels of caspase-3 in associated with neuronal marker NF200, and same things in we could see in cerebellum. So in long story short, I do not believe that their mode of action is neuron specific, is broad neuronal, causing damage in vast majority of neurons we have seen.
So I guess how do you in Parkinson's disease, we do see heavier degeneration in the nigrostriatal system and maybe locus coeruleus and olfactory bulb as well, which I think is interesting because usually it's been actually suggested that things kind of start in the olfactory and spread the other direction rather than, it's interesting that you're injecting it in the midbrain striatum cortex and it's kind of going the opposite direction to what's been.
Why, you mean?
Yeah, which is super interesting. But how do you think about, you don't see widespread degeneration in the cortex in Parkinson's disease. So do you think that there's some kind of a reason?
There is. That is another point I wanted to make, but I haven't succeeded. It seems so. In reality, Parkinson's disease, because of its motor symptom and the first sign that it's being diagnosed by motor symptom, it is believed to be more brainstem and dopaminergic neurons. But in reality, there is massive neuronal degeneration in different areas, including dopaminergic neurons. But perhaps dopaminergic neurons, as it has been suggested, they are very well wired and they required more energy than other maybe types of neurons, and that is the reason that they are showing more vulnerability to damages or oxidative stress and all other sort of dysfunction intracellularly. But at least the mode of action of damaged mitochondrial DNA do not seem to be neuron specific as such.
Interesting. I do see a hand up from Andy. So Andy, if you want to go ahead and ask the question.
Yes. So one of the things I'm interested in, kind of along those same lines, is the presentation of patients is highly heterogeneous, and as you mentioned, the motor symptoms is the key diagnostic point, and then there seems to be a range of dementia for some Parkinson's patients and not others. Given your data here are in these interferon knockout mice, do you think that the interferon deficiency might be kind of the defining molecular feature that drives whether some patients progress to dementia or not?
Most likely it is one of the pathways at least associated. The previous study we had was showing very strong association with PD in general, but also it was much more dysregulated as patient progressed to dementia. And in mice we see similar, but one thing again to make clear, so the mitochondrial DNA deletions and mutations we see 6 weeks of age and in different regions, but quite significantly in midbrain. So it seems that the dopaminergic neurons are somehow more sensitive to any type of disturbances, including lack of type 1 interferon signaling. And but this is being anyhow progressed. I think in patient also it is becoming more and more clear that non-motor symptom might have started much earlier, but it hasn't received the diagnosis because of the lack of yet diagnostic marker and the motor symptoms and so on. So what is hen and egg here? I'm not sure it is clear, but at least in my mind, the major pathology start when any sort of mitochondrial hemostasis is being disturbed. Now, what is triggering it and in which area might impact? As we know, actually, quite many viral infection has been associated also with loss of olfactory bulb as first sign or symptom of Parkinson's disease as well. So I have a strong conviction, but no enough evidence that that lack of type 1 signaling or any viral hemostatic signaling might be associated actually with the neuronal mitochondrial pathway. And like even COVID-19 was strongly shown to have impact and progression of some of the symptoms of Parkinson. So it can be many different, as you mentioned, heterogeneous pathway, but I think they are all somehow getting connected to mitochondrial hemostasis.
That's really interesting. I think one, and that was actually a question I had as well, which you sort of addressed here, which is, where do you see the convergence for some of the other, maybe known causes from the genetics that we know, synuclein, obviously, and there's been a little bit of literature on synuclein and TLR4 kind of separately from what you've shown here as well, and maybe some of the kind of less intuitive, maybe some of the risk factors like GBA or pink Parkin, obviously, which do overlap with mitochondria. You see this obviously as the area of convergence, right? Can you speak maybe a little bit more to that?
So proteinopathies in general, alpha-synuclein as a part of it, are also causing disturbance in mitochondrial hemostasis. We are not working particularly with alpha-synuclein pathology as such, but we see quite often aggregated phosphorylated protein in this bundle of Lewy body. They are quite somehow coming close to the mitochondrial hemostasis somehow in some way. And it was a beautiful study showing that they might be actually involved in the formation of the Lewy body as well. So somehow it seems that if mitochondrial energy production is disturbed, now is as a result of proteinopathies, or it causes proteinopathies, they are very, very well converged at some levels.
Thank you. We do have a hand up. I am going to butcher the name. Unless you want to say it yourself, that would be great.
Nice. Yeah, thank you. I have a question about, and I'm not in the field, so this might be a naive question. What is the mechanism by which interferon signaling induces mitochondrial DNA damage?
Yes.
Is this known?
I'm naive in that too. So it is something that my lab is very interested and we are trying to understand. So this is not a previously well-described pathway, but at least we believe that interferon signaling, it is important for neuronal hemostasis in different levels. And this is because of also its polyotropic function. So one way by which it's important, it is regulating mitochondrial fission, it's regulating mitophagy, it's regulating autophagy, and all these are important for removal of damaged mitochondria and damaged mitochondrial DNA. So that is at least one way by which we believe it is lack of the upstream of mitochondria or mitophagy can contribute to accumulation of excessive oxidative stress and damaged mitochondria intracellularly. And perhaps I do not believe this is an alternative to mitophagy, but it is a response to toxin that it is cells do not want to accumulate, then they expel it out in extracellular vesicle or directly. So it is, at least in type 1 interferon, it is quite important for upstream of mitophagy, and that is one way by which it is doing its job. But it is not excluding the possibility that is also involved in mitochondrial DNA replication, repair, and so on. That is something of interest, but we don't know much about it today.
Okay, thank you.
Thanks for the question. We do have lots of questions in the chat as well, so I'm just going to try to go through them rather quickly. Young Heck asks, thank you for your presentation. Have you checked other mitochondrial complex function or complex related protein expression, and specifically asked about complex 3, which is closely related to ROS production?
We are looking at them, but from top of my head, I do not recall something about complex 3 is jumping out. I'm afraid I can't answer you well today, but hopefully we will know more about it soon.
And I want to tack on a question to that too. What about mitophagy specifically, particularly pink Parkin-mediated mitophagy? Have you looked at that?
Yeah, we are looking at it also. We hope to have some answers soon as well.
Would you, and I'm selfishly asking this question because we are looking to enhance mitophagy with our molecules, but would you expect that enhancing mitophagy would alleviate some of the negative effects that you're seeing?
Sure. So it depends where in the pathway it works. That is because we have some evidence that mitophagy enhancing agent might work, but not completely, because it depends if it is since type 1 interferon pathway is upstream of the mitophagy. Somehow we can maybe circumvent it, but not always. It depends where in the pathway the agents would act.
Right. We can test that and find out. Would be good. Another question is RSP3 a ribosomal protein? And if so, did you look if translation of efficiency is affected?
Translational efficiency of nuclear DNA or mitochondrial DNA, perhaps, but that is basically something I was trying to address in response to previous questions. So it's something of interest to us, but we don't know the answer yet today.
And I think there was another question that you've already answered through other questions, so we'll skip that one. Kayla asked, could the findings here have broader applications to other neurodegenerative diseases beyond Parkinson's disease? And I'll add to that as well. Have you looked at brains from other neurodegenerative diseases, and do you see kind of a similar pattern, or is it different?
Indeed, when we started working with transcriptomic analysis, we have also looked at AD, and we see quite much of overlap in dysregulated pathways. But we haven't had chance to look at particularly at other cohorts of patients with AD to develop further. But one thing of interest, maybe I did not have chance to highlight, is that this mice that lack type 1 interferon, they develop, among the proteinopathies that they develop is also phosphorylated A-beta, and they do have also A-beta plaque, as we have published it in Annals of Neurology 2021. So they have quite much of overlap in disease pathology when it comes to PD and AD.
And so what do you think is the factor that then pushes a person towards one versus the other?
Yeah, that's $1 million question, but I just think that it depends on perhaps where the sensitivity or the pathology hits. For instance, what type of maybe infection, what part of the CNS or associated pathways being triggered, it can cause vulnerability in certain area first and second, and that is how the pathology maybe develops. That's just completely hypothetical.
Yep, makes sense. I know I asked the hard question, but those are kind of the fun questions to think about.
Yeah, I completely agree, and that is perhaps so in the way I am looking at neurodegenerative diseases, actually quite all Parkinsonian that are in one category together also with Alzheimer, they share quite heavy burden of pathology and clinical symptom. That's why sometimes even they are being shifted from one category to another, depending on what more they develop and what drug they do not respond to. But in reality, I think if we try to understand molecular paths that are leading to neurodegeneration and being able early on to address some of this pathway, we might be lucky to actually hit many of these diseases that are by end converging in reality.
Makes sense. I think you sort of touched on this already, but Jody had asked about the mechanism by which the spread of the PD pathology goes to the distal areas of brain, and you talked about this a little bit already, but I don't know if there's anything you'd like to add about the specific or maybe what you're doing.
I'm aware of the fact that it has been hot topic that maybe even earlier was how it is spreading through the brain and then from periphery to brain or vice versa. I have no evidence to believe that one or other might be the way, but in reality, central nervous system, although believed to be completely isolated organ, it's not. It's associated with the rest, and where exactly the start and the progress, I cannot convince anyone today, but at least the study we have done, we have shown that a pathology can be triggered by damaged mitochondrial DNA and can spread to other areas, and that is behind the progression. But if that is coming from blood to brain or brain to blood, it is something that I think next 10 years, perhaps we will have much more clarity in it.
Definitely. And we talked about different neuronal types, but I guess the other obligatory question is other cell types within the brain, maybe microglia, astrocytes.
That is a very, very interesting question too, yes. You are right. So other cells are also might be involved, but at least the pathology we have seen, it's neuronal. So if we knock the type 1 interferon in astrocyte, they do, this is actually some unpublished yet, but they do mimic some neuropsychiatrical outcome. But the neuronal lack of type 1 interferon, basically it is mimicking the most pathology and clinical outcome. So definitely one can take the conclusion that the combination of cells and pathology in different cells are contributing to disease. But at least we haven't seen major evidence that, for instance, microglia are among the main disease causing. Even in this study that I mentioned, when we take damaged mitochondrial DNA and put it in the brain, you expect that microglia are among the cells that they should uptake it and get activated and be involved, but we couldn't see much of microgliosis. But astrogliosis, yes, we could say.
Interesting. Not what I would have hypothesized, but that's very interesting.
Yes. Yes. So why I am not so sure. Maybe if I could just hypothesize from immunological background, for me, my microglia are actually the scavenging cells that they are there to clean up, and what we cannot take care by autophagy, mitophagy, and spit it out, actually they are there to clean. So per se, I do believe that they are more involved in helping to remove the pathology than causing. But of course, if something goes completely wrong in them, they would also contribute to disease. But in this model, we couldn't see much of microglia activation, but astrocyte activation.
Interesting. Different question from Anahid. And I know we talked a little bit about mtDNA potentially as a biomarker, which I think we're all very excited about because it can help with diagnosis, early diagnosis, patient stratification, and then progression as well. But she asked specifically if you've looked at RPS3 protein as a potential biomarker as well.
Not yet, not yet.
Would be
But
interesting.
this is a very, very interesting, actually, maybe a biomarker. I am not so sure how easily accessible it is, but it should be optimized and studied definitely.
And then she asked also about, is there some kind of regulatory feedback loop that kind of manages the interactions between the RPS3 and TLR4? And I would even take it one step further and go, between the initial inflammatory processes that you see in mitochondrial DNA.
This is something that we don't know. I mean, this is basically, I believe, the first study to show that it is associated with damaged mitochondrial DNA and extrusion and uptake. So more than this, we haven't been yet able to identify additional pathways by which it might be important, but surely they require, I mean, cells require RPS3 for handling it. But also it can get involved in the pathology.
Yeah, it makes sense. I think that's it does kind of get a bit more complex because I think these regulatory loops often do exist, and so just kind of trying to figure out where the starting point in the loop is often a major challenge.
Completely agree. I mean, we cannot exclude that any defect in RPS3 function might cause the same loop to basically the pathway to crash as well, but it's not something we have been working with yet. The only thing that I showed were blocking the RPS3 or knocking it down to see what can do. But if there are mutation, if there are loss of function or gain of function, somehow it's not something we know of today.
And it has not been reported in the literature.
Nothing I know of, nothing I have seen.
Yeah, I hadn't seen anything either, but I thought maybe I'd missed it. That's interesting as well. Would be maybe something the geneticists can have a look at kind of retrospectively and see if there's anything there.
Yes, sure.
Speaking of genetics, I do wonder about this as well. One of the, in thinking about kind of the spectrum of Parkinson's to Alzheimer's and PD with dementia kind of in the middle, the Parkinson's patients with pink Parkin mutations generally don't have a lot of dementia component to the disease, and they're kind of slower progressors than the rest. But obviously they have the mitochondrial deficits, right? That's that's the primary cause in their case. Do you have any thoughts on what might be happening there?
Yeah, that's a very interesting question. We were also quite wondering when also searching literature, writing the paper, because I mean, all three knock out of associated genes with mitochondrial function also do not develop any disease pathology, interestingly. So, but lack of interferon does the job. So it seems that maybe that is actually the reason, because this is sort of a signaling that has polyotropic impact. So it does do many damages at different stages of cell processes. But mitochondria has plasticity as well. So if one or another is missing for quite long period of time, they can cope with it. That's why also in general, PDs or neurodegenerative diseases are the diseases of aging. So we have been able to cope with it for quite many years.
And actually, that brings me to another question that was just here, which is the aging component. Do you see, if you've looked at Well, and I know with the mice, you had some 12-month-old versus younger mice. And I'm curious if you've looked in healthy humans, like young versus old at all. But what do you think about the aging component just by itself outside of the neurodegenerative disease?
No, we haven't had this luxury to do a study healthy aging. But yes, mice. So when we are looking at mice for PD model, we always have their corresponding wild type, and surely by aging they also start to develop some sort of pathology. Like after 18-20 months of age, they already have some sort of proteinopathies, for instance. So aging has definitely impact in even normal progress of brain function, but we haven't studied it in human. But again, in mice of Parkinson model, they develop damages in mitochondrial DNA much earlier than any other pathology or symptom. So basically 6 weeks of age, we already see heavy mitochondrial DNA damages when there is no any proteinopathies, no any clinical outcome, nothing. So it is at least a strong evidence for us that mitochondrial damages, mitochondrial DNA specifically damages, could be preceding the pathology. But what is causing it, it is depending on different, I think, condition. But that's another interesting thing about type 1 signaling. It is, at least to date, we know more than 200 some genes that are involved in this. So, and that is one of the reasons we believe this pathway is not easily coming up, because with GWASs, we look at individual genes and mutation in genes, but any single defect in any of these protein can cause the crash of the card house, basically.
Right. And maybe in different ways too.
Maybe a little bit differently as well, but even maybe similar, but they are not showing up because one component in one patient and another in another. So we should find better ways by combining the accumulative or cumulative effect of a pathway rather than one gene.
Yep, agreed. And I think these this is partially why we're big on the biomarker thought, is if we can look at different patients and look at the heterogeneity of the patients and see which ones are kind of falling into which buckets as far as the categories. Yeah, makes a lot of sense. Great. Well, I don't know if there are any, I think I'm not sure. Oh, there was one more question from Kayla. Can interventions be developed to protect the mtDNA before it's damaged? I think we talked a little bit about potentially going after mitophagy, which is, after that point, possibly. Well, could be before as well.
One way by which, but yes, also understanding in general the mitochondrial DNA replication and repair. I think it is important in this, because they might be correlated, but they might be independent entity as well. That any of them goes wrong can again cause same collapse. Right,
makes sense. All right. Well, I think if I missed your question, I apologize if it's out there. Is there any actually, I think we're pretty much out of time, so we'll be right on time. Thank you again so much for taking the time to share with us, and I appreciate that you did it even though you had a cold. I hope you feel better. Yeah, and we look forward to seeing more of this really interesting data on coming out of your lab as well.
Thank you very much. Thank you for the opportunity and for meeting you with your interesting question. I enjoyed it.
Likewise. Bye-bye for now.
Bye-bye.